Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 5.2, meiosis and genetic diversity. In topic 5.1 we discussed meiosis and now we'll be looking at all of the ways that meiosis contributes to genetic diversity. We'll be looking at crossing over, we'll be looking at independent assortment, and we'll be looking at fertilization of gametes. And our suggested skill, questions and methods, 3A, identify or pose a testable question based on an observation, data, or a model. But before we can look at how meiosis increases genetic diversity, we have to ask ourselves a big question. Why do we actually want genetic diversity? What is the benefit? And so when we think about a diploid organism with diploid cells, such as humans, there's going to be two alleles per gene. And one allele is going to be inherited from the father and one from the mother. And the allele is just a type of that gene. So for instance, when talking about, say, eye color, your mom could have brown eyes and you would inherit a brown eye allele. That's the form of the gene that you would inherit from your mom, while your dad could have blue eyes and you would inherit that allele of the gene. And this contributes to the genotype or the genome of these organisms, the genes themselves, which is then going to show in your outward appearance or the phenotype. And sexual reproduction causes these alleles to be mixed up and produce varying offspring. You're not going to look exactly like your mom or like your dad. You're going to be a mix of the two, and that's because your genotype is a mix of the two. And if an entire population consisted of organisms with the same genotype, which would then result in the same phenotype, there's a high chance it would not be able to survive any environmental changes. Especially because if the change affects one organism, that means it could affect all of the organisms because of how similar they are. A good example is, and one that gets brought up quite frequently, are moths during the Industrial Revolution. And these guys sat on the trees, and before the Industrial Revolution, the bark was fairly light. So the moths that were able to camouflage themselves and survive best were the lighter and whiter moths. However, once the Industrial Revolution began and smoke and soot began pouring out from all of factories all over England, a large amount of these trees became covered in ash and soot and turned a darker color, which then meant that the moths that were able to survive better now were the ones darker in shade. Now, if all of the moths were the same color, this environmental change, especially if they were all the lighter shade, would cause the moths population to be wiped out because now they have no source of camouflage. They could be easily found by predators. So by having genetic diversity within a population, there's a higher chance that the population as a whole will survive because different organisms are better adapted to different environments. So now that we've discussed the benefits of genetic variation, let's talk about how meiosis increases genetic variation within a population, going straight from its beginning all the way to fertilization and sexual reproduction. So first, of course, the DNA is going to be duplicated during synthesis, and right at the beginning of meiosis, during meiosis 1, during prophase 1, crossing over is going to occur. So we have our paternal chromosome made up of two sister chromatids and the maternal chromosome made of two sister chromatids, and these came from S phase, and they're going to join together in a tetrad, and then a thing called crossing over is going to occur, where a piece of paternal chromosome is going to swap with a piece of maternal chromosome. And what this results in is now we have four sister chromatids that are completely genetically non-identical, completely genetically different. And now, as we continue through meiosis, now pretend these are still attached, we reach metaphase 1, where these tetrads have lined up at the metaphase plate to be separated. And when these tetrads are at the metaphase plate, so let me draw the metaphase plate, and let's copy over this one, for instance. So we have our paternal chromosome on this side and maternal chromosome on that side and pretend they're joined together, and actually this one's a little blue and that one's a little red because crossing over has happened, what's going to happen is that during anaphase 1, when these homologous chromosomes are pulled apart, the maternal chromosome is going to end up in this daughter cell and the paternal chromosome is going to end up in that daughter cell. But remember, these aren't the only chromosomes in the human cell. There's going to be 23 pairs of chromosomes in the human cell. And what can happen is that they don't always need to line up this way. Sometimes the maternal chromosome can end up on this side and the paternal chromosome can end up on that side. And there are essentially two options to how this happens. And so there's a 50% chance for each daughter cell to gain a maternal or paternal chromosome, which creates a lot of combinations. And then during meiosis 2, when these chromosomes, the two sister chromatids, decide to separate during anaphase 2, then we have two genetically different sister chromatids because one has had crossing over happen to it while the other is pure maternal or pure paternal chromosome. So at the end, you're going to get 4 daughter cells that are all genetically different. And now going even beyond this, even beyond the creation of unique eggs and sperm, when these combine, especially because each one is unique during sexual reproduction, you're able to create 70 trillion genetic combinations because you're now multiplying the genetic probability of a single unique egg from one parent and a single unique sperm from the other. And so there's so many combinations that can happen that increase genetic variation within the population. So there you have it. Genetic diversity, good. Meiosis makes it happen. We'll see you next in 5.3 to talk about a guy named Gregor Mendel.